A breakthrough in COVID-19 detection. Sounds interesting. Let me ask Dr. Khubaib tomorrow about this.
Good morning, class. Today's lecture will be on microbial sensors. We also have a guest lecture speaker, Dr. Ming. Before we begin, does anyone know how microorganisms help in detecting certain issues? For example, in environmental or in human body?
Doctor, I came across an article yesterday about a breakthrough in COVID-19 detection. Is this topic relevant?
Yes, it's very good. The microbial sensors is the concept behind the virus and bacteria. So, microbial sensors are analytical devices that use microbes. They detect specific microbe substances by harnessing microbial activities. These activities are then converted into measurable sensors. Measurable sensors signal. The sensors are widely used in diagnostic, environmental issues, food technologies, and biotechnologies, offering precise and efficient detecting methods.
Professor, are there different types of microbial sensors?
Of course, there are various types of microbial sensors. Electrical sensors detect electrical signals. Opticals one major high basic changes. Thermal sensors sense heat produced by the microbes, while piezoelectric types detect mass and stress changes. There is one more, pulsar sensors using genetically engineered microbes for specific detection. So let's invite Dr. Ming to explain more about how they are applied.
Hi everyone, let us continue with the application of microbial sensors. First, we use genetically modified bacteria to help recognize and generate a signal in response to a molecule of interest. For example, we have the recombinant BIOMEDICAL cells to detect arsenic. Next, we have the microbial few cells, or MFCs, to help as optical transducers. These MFCs have higher sensitivity and selective sensing capability. So next, we also have the fluorescent microbial sensors to help emit fluorescence upon detection. So, for example, we have the recombinant yeast to help emit fluorescence when exposed to genotoxins. Next, we also can use microbial sensors for healthcare and biomedical use. We can use these sensors to detect bacterial pathogens, signal the presence of said pathogens, and diagnose the infections. For example, we've had acyl homocysteine lactose or BHLs to use as biomarkers for these infections. And next, we have the photobacterial nanopermeases to detect urinary tract infection. And lastly, we have the E. coli SOS response to detect DNA damage.
Does this mean that we can use the same method for bioremediation and other industrial fields?
Correct. Would you like to share your understanding on that?
Yeah, sure. We have two more applications of microbial sensors, bioremediation and industry of food and beverages. In bioremediation, the microbial sensors can be used to detect environmental pollutants like metals, petroleum hydrocarbons, and pesticides. The pollutants produce measurable signals that the microbes can detect. Monitoring bioremediation efficiency as sensors can track microbial respiration of enzyme activity and real-time monitoring of microbial sensor activities. It also allows for continuous monitoring of the pollution level, and it's very cost-effective and time-efficient. For the last application will be in the industry of food and beverages. It can be used to detect pathogens or spoilage microbes using bacteria or yeast. It can be used to monitor fermentation by measuring ethanol and CO2 and lactic acid levels, and it can also detect food additives and residues.
Great! Thank you, Shui and Abdulrahman, for actually participating in the class. Let's take a look at the videos from NTU Singapore to explain and share their development of the latest breakthrough in COVID-19 detection.
Hi everyone, my name is Viken and together with me is Chen and we are from NTA Singapore. Today we are going to share with everyone a glorious invention which is the crystal model. So our development is called the Viken CRISPR Cas12a and Cas13. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Viken is the variant nucleotide drug. So our development is mainly targeted on the SARS-CoV-2 virus, which is commonly known as the COVID-19 virus. Our development works on the principle of DNA or RNA cleavage, which is done by the Cas12a and Cas13 enzymes, and it is guided by the gRNA, which is a preserved fragment from the predecessor DNA method. Our development correlates mutation in a way that it can detect and identify the presence of that virus, even though it has undergone various multiple mutations. For example, we are able to detect the presence of COVID-19 in that patient, even though the mutated is infected by the mutated variant, which is the Omicron or Delta. Our development is able to detect variants of the SARS-CoV-2 virus, such as the Omicron and Delta as mentioned earlier, in a very short period of time. So test kits provided that you can see in the market, they contains an assay which is a solution in a small bottle. It contains Cas12a and Cas13 enzyme, which allows visual interpretation of natural flow stream, which is the result that we see. So the result is called as colorimetric result, where the single line integrates, single line shows negative result and the double line shows positive result. And it is very portable and suitable for using airports, CPDs, and pharmacies for rapid tests. New test kits are available in the market, which they can test multiple viruses in one test, such as influenza A, influenza B, COVID-19, and many more.
That's right. Now let's look at the benefit and the downside of this microbial sensor. Firstly, the benefits of the microbial sensor is that they can detect a very low concentration of analytes and are highly specific. Second, they also provide a real-time monitoring of analytes, and this enables the scientists to make a rapid decision. Furthermore, they are often less expensive compared to the traditional analytical methods. Despite the benefits, there are some downside too. Microbial sensors may be unstable due to some external conditions like pH, temperature, or oxygen level changes. Also, they might give wrong results sometimes as the other substances in sample might affect them. Lastly, the use of the microorganism in the biosensors is often controlled by some strict rules, especially in food and medical application. Right. That's all for our first. Thank you.
After watching the video, can someone share what are your thoughts on the future aspect of microbial sensors?
Me, I think future aspect can be digestive health, blood monitoring, or even a personalized medicine. It's like digestive health, it can detect the gut disease or inflammation disease, which monitoring the colon bacteria health. In the blood monitoring, it can detect the disease like cancer of our bloods or even like high blood or low blood pressure. And in the personalized medicines, it's like can put in the contact lens to detect the infections. And next is the skin patch to detect the sweat chemicals.
Yes, your idea is on point. It seems like everyone has understood the concept of microbial sensors. Very good, very good. Class, that's all for today. See you all next week. Bye.